We got the OGs here. Okay, you guys. Let's start going through these. See how many we can get through all the way. So let's see. Yeah, some of this I'll probably have to pull up the book and we can go through some of it, and some of it is more like knowing the actual process. Other parts of it is more just like a little bit of memorization. So, okay, question 1. Almost all the oxygen a person consumes during breathing is converted to which of the following? Acetyl-CoA, CO2, H2O, or all of the above?
H2O.
Yeah, the answer is, I'll do my highlighter, is H2O. All the oxygen consumed during breathing is converted to our H2O. Let me pull up real quick the...
If anybody hears snoring in the background, it's just the dog sleeping next to me.
So here's kind of our...
blame it on the dog.
our electron transport chain. So our everything flows all the way through. And then at the very, very end, we get converting of everything to H2O. Where do we produce carbon dioxide? In all the steps of creating energy, where do we produce carbon dioxide?
Crickets.
I mean, we create some carbon dioxide in TCA, right?
Yeah, the TCA cycle, right? So we're producing that carbon dioxide within the TCA cycle. I'm going to give you a little bit of extra information here. This is going to be later on stuff, but next trimester and try 2 and a little bit in try 3 as well, you'll talk about, and I think I mentioned this before as well. You have the bicarbonate buffer system. Remember hearing about that? Vaguely, yeah. You don't need to know it right now for this material, but it's just kind of letting you guys know what's going on. We produce carbon dioxide, especially in the citric acid cycle, in the Krebs cycle. And carbon dioxide, because it's acidic, we need a way to transport it properly in the blood without messing up with our pH. So we have a buffer system where we turn the carbon dioxide in order to get it into the blood and to transport it. We turn it into bicarbonate. And so there's a kind of little shift in a game that our body plays to go back and forth between CO2 and CO2 and bicarbonate in order to transport the carbon dioxide through our blood until it gets to the lung, and then it converts back to CO2 inside of our lung and gets switched out and replaced by oxygen. So that's something you'll go into a little bit deeper in Phys 1, as well as a touch. I think they touch on it in Biochem 2, as well as you'll want to remember this a little bit as well for systems pathology in Tri 4. You'll talk a little bit about the bicarbonate buffer system and kind of what sort of diseases and stuff can mess with that. So just wanted to mention that in there real quick. Okay. Antimycin A blocks electron transport between which of the following? Cytochromes B and C1, cytochromes S, AA3, and oxygen, NADH, and coenzyme Q, and cytochrome B.
B and C1.
The answer is B and C1. Am I saying? If I can find a good picture. He has a decent picture in his book. This is one of those questions where they won't ask a ton like this. If he asks a question like this, it's gonna be this exact question. The cytochrome B and C1. Nope. Wouldn't not super concerned about this. This is more just if you see this question a couple of times, come back here and read back through it before you take the test. You probably have this exact question on the exam. ATP synthetase in the electron transport chain contains two functional units. One of these is an electron transporter, or all of the above are true, a dehydrogenase component, or a transmembrane protein channel. Well, when we talk about the electron transport chain, right? Maybe we should start with this before we dive deep into it. Open with this. And 2C cycle, sweet. So we've gone through all of our citric acid cycle, all of this sort of stuff, all of our Krebs cycle. We got all the way through. Main goal of our citric acid cycle was to produce all this beautiful stuff called NADH and a little bit of FADH2, right? We have all of those amazing fun steps. And then we finally get here to the electron transport chain. This is where our big payoff of all that energy is going to come from. I want to find stuff we can come back to and review. This guy. Right. So we have our electron flow that we're going to be having. With all of this, how do we feel roughly right now about the electron transport chain, about this, the general process of it? Like on a scale of 1 to 10, how do we feel about the process?
Like
Not great, but not horrible.
Not great, but not horrible. Okay. Yeah. So what we want to understand is the biggest thing that there's. We'll look at the main big components, okay? We have the different complexes, complex 1, complex 2, complex 3, 4, and then 5 with our ATP synthetase chunk as well, okay? The big things that we want, also coenzyme Q. So with the electron transport chain, literally the name is telling us exactly what's happening. It's an electron, so it's electron transport, so it transports the electrons through a chain of different proteins. It's electron transport chain. So that's its main job and its main goal is to move the electrons down down the membrane. The reason why this is important, if we think about electrons moving through something, imagine thinking like a wire, very, very similar concept. As the electrons move and pass through, it gives energy. It allows there to be energy to be able to do certain actions, just like how we get electricity and how we power everything with electrons moving through wires. Similar idea here, except the action that we're trying to perform is pumping hydrogen atoms from the mitochondrial matrix into the intermembrane space. Okay? Going from that mitochondrial matrix into the intermembrane space. So each of our different complexes has a specific job and different components and help us to transport these electrons. So complex 1. Everything starts here in complex 1. where we have our NADH. And if we think way back a little bit to remember when we're talking about oxidation and reduction reactions, right? When an H gets put onto something, what is the electrons are coming with that H. We are using the hydrogen bonding with the electron and attaching it there in order to transport the electron. Not just the hydrogen, but mainly the electron is what we're trying to transport. So we built all of our NADH, our beautiful NADH, from our citric acid cycle. And now at this point, we want to use those electrons, those high energy electrons, to be able to pump the hydrogens across and to create a gradient. So complex 1 is an NADH dehydrogenase. What do dehydrogenases do? What's their function?
Remove hydrogen.
Yeah, they remove hydrogen. So literally what's going to happen is our NADH is going to come, it's going to bind onto our complex 1, and it's going to rip off that hydrogen, and the electron that was connecting with that hydrogen is going to get sucked into and pulled into complex 1. So complex 1 is holding that electron and it's moving it from where it took it from NADH over to another enzyme called CoQ. Now CoQ, and we'll see this in a little bit with questions, CoQ is probably one of the most asked about in this next chunk because it is very, very important. And you'll also see, this is also a board's question, that CoQ10 is most similar to vitamin K. So vitamin K is important because it's very similar to CoQ, and we need that for our electron transport. Okay. Within this process of pulling off the electron and this electron moving, right? Complex 1 is going to use some of that energy to pump hydrogens up through the membrane. Okay. So we get an electron coming off, electron transported, given then to CoQ, and hydrogens being pumped up through. We then have complex 2. Complex 2 is a slightly different job. It's basically doing the exact same thing as complex 1, minus 2 things. 1, it gets the FADH2 instead of NADH. And it also does it because it's not a transmembrane protein, so it doesn't go all the way through the membrane. It doesn't actually allow hydrogen to pass from the mitochondrial matrix up to the intermembrane space. Other than that, it's the same exact job, just with FADH2 instead of NADH, where it rips off it separates the hydrogens with a succinate dehydrogenase. Again, dehydrogenase breaks off the hydrogens. And it steals the electrons to bring them up and deliver them to CoQ, or coenzyme Q, or also known as ubiquinon, because we love our AKAs. So this is how you'll see it. You will see it as coenzyme Q, CoQ, or ubiquinon. Okay. So the main function of complex 1 and complex 2 is to rip off the electrons and transport them to CoQ, aka ubiquinon. From there, from ubiquinon or CoQ carries those electrons over into complex 3. And complex 3, kind of like how we talked about with G protein coupled receptors. Remember, we think back to G protein coupled receptors. We had like the whole big G protein, but the G protein was made up of gamma, alpha, beta, and GDP. How it had those different parts. Remember that?
Yes.
Yeah.
It's very, very similar idea here. where we have complex 3, and part of making up complex 3, we have these different cytochromes. Within complex 3, we have cytochrome B, and we have cytochrome C1. And again, their job is just, they're inside of it. They're used to help us transport the electrons. Okay. That's kind of the way he'll ask you questions on those, is kind of associating either where does the electron go next, or let's say this specific cytochrome was not present for whatever reason, where would the electron stop, or what would not occur, those kind of questions. Just knowing where cytochrome B and cytochrome C1, again, cytochrome C, cytochrome A, A3, all of these guys, where they're located specifically, that's the main thing that you want to know, is where they're located, so in which complex they are located in. And if there's anything specific that's going to be blocking them, where will the electron come from, or where will the electron go in a normal pathway? Those are the things he's going to ask about. Just like complex 1, complex 3 uses the energy from the electrons to pump hydrogens up through, and then it continues down the chain and passes it to cytochrome C. Cytochrome C then passes the electron to complex 4. Complex 4 has cytochrome A and cytochrome A3. Again, same exact job. They have they're there. They make up part of the complex 4. They pass the electron along, and the energy from the electron pumps up more hydrogen. And all of this hydrogen getting pumped up and up and up into the intermembrane space creates a chemical gradient. Chemical gradient, all that means is that we have a solution, right? We have a barrier in between, and we're getting a bunch more stuff on one side compared to the other. Kind of like this. So following all the laws and stuff that we've learned, if we create an opening, what's going to happen to all of this material on one side if we open it up?
Gonna try to find equilibrium.
Exactly, they're going to try and find equilibrium. So all of these hydrogens, all of this stuff that's over here is going to move. And when things move, they create energy. And that's what we want to take advantage of. So we've built up all these hydrogens over and up and up and up and on this whole, on one side. And we finally finished with our electron here in complex 4. And so we deliver the electron to an oxygen and hydrogen to create water. And now we use this chemical gradient with all these hydrogens here. We place a tube, or an intermembrane protein, an intermembrane channel right here. That is a free spot for the hydrogens to just slide on through to be able to make that equilibrium, to reach equilibrium. So that way it's supposed to be an even number on both sides so that things are balanced. This movement of hydrogens through here, you can kind of think of it, I like to think of it like if we were to make a cut right here and then kind of turn it. So we're looking down inside of it. The way that this is super simplified, but the good way to think about it is there's essentially like a wind turbine or like a water turbine inside of here that as the hydrogens flow through, it pushes it and makes this spin. So it's a mechanical movement, so it makes it spin. It's almost like, think of like water flowing through a dam turbine. It makes it spin. And that spinning movement energy, so we're taking a chemical gradient, potential energy, turning it into movement energy, and then that movement energy, we're going to use it to convert it to chemical energy, to bind a phosphorus onto an ADP to create ATP, adenosine triphosphate, which is our main source of energy in the body.
The
Up
way
to
we
that point
talked
is
about it, I was gonna say the way we talked about it on Friday in Dr. Sarkar's one-on-one group. Breathe in, breathe out.
yeah, so we have all of this material. Okay, now having gone over it now, does that sit a little bit better? It still looks really confusing,
Yeah.
but this is one of those things again, just like the citric acid cycle, just like glycolysis. Draw it out a couple of times and simplify it down. Like this looks way more confusing than it needs to be. I would even like we have membrane, complex 1, 2, 3. And then kind of like this. I haven't drawn this out in a long time. So there's 1, 2, 3, 4, and there's 5. Then the first circle right here. What's this enzyme? What's this one called?
CoQ.
CoQ. What's another name for CoQ?
Ubiquinone.
Yeah,
That's it.
ubiquinone.
Ubiquinone.
Then we get over to complex. Oh, let's keep going here. Complex 1. What does complex 1 break apart?
NADH.
Good. So NADH goes into here. What else does complex 1 do?
Transports 4 hydrogens across into the intra mitochondrial membrane.
Yeah, we get the hydrogen movement. Perfect. I don't think he usually asks if like the number that each one moves, just that know which ones move and which ones don't. Basically, all of them move hydrogen up except for complex 2. Usually he doesn't ask about the numbers, if I remember correctly. Okay. Complex 2, what's complex 2's job? What does it break up?
FADH?
FADH.
Or FADH2.
Yeah, FADH2. It grabs electrons. Now, both of these, complex 1 and complex 2, what does it do with those electrons? Where does it pass them to?
Takes them to CoQ.
Bam, passes them to CoQ. What does CoQ do then?
Sends them to 3.
Moves it to 3.
Transfers them to
Passes them to
complex
3.
3.
Transports them to complex 3 because it's a chain. We went 1, 2, and now we're down to 3. Now 3 is made up of a couple of different parts, right? You remember which parts are made up in complex 3?
Cytochrome
cytochrome
C.
B. Oh,
B, yep, B and
Oh, B, yeah.
C1.
1.
Yeah. Our electron goes through. What else? As the electron passes through cytochrome B and cytochrome C1, what else does complex 3 do?
Moving
Transports
hydrogens.
hydrogens.
Yeah, it moves hydrogens. What's next? Pass electron to what?
Cytochrome C.
Cytochrome
Cytochrome-
B.
Cytochrome C. This one. So cytochrome B was in
Or
complex
C.
3. Now cytochrome C. Where does cytochrome C transport it next to?
4.
4.
Into complex 4. Complex 4 is made up of A and A3. And then finally, where does our electron go to?
Make water.
Makes H2O. Sweet. What else does complex 4 do?
Moves hydrogens up.
Yeah, pumps hydrogens. Conceptually, if we think about it, does complex 4 pump out less or more hydrogens than complex 1?
Plus.
less. Why do we think that it pumps out less?
The concentration the concentration by the time it gets to that point is already less, so it doesn't have as many to give.
So there's one reason. An
Well,
even
you're
bigger,
making
an
water,
even bigger
so
reason
you
as well.
need hydrogen, right?
Think about the electron, right? The electron has a certain amount of energy that it's able to give. It's able to pass on. And here at the very, very beginning, in complex 1, it has a lot of energy and it's very, very high powered, right?
Mhm.
And then even here in complex 3, we're getting fresh electrons from FADH2 as well. So that's going to increase the amount of power that it has to pump up hydrogens. But once we've already used some of the energy here in complex 1 and use energy here in complex 2, the electron has less energy to give, so it's going to pump less hydrogen as well. So because there's increase in concentration, because the electron is lower energy, both of those reasons are why complex 4 doesn't pump as much hydrogen. Okay, so now we have complex 4 finished. We've now have made water. Now what? What happens next?
The hydrogens start to trickle back down.
They start to trickle down back through right here. What is this called right here? An intramembrane what?
Pump.
Yeah, well, it's our ATPase. It's our ATP synthetase. It goes through an intramembrane channel. Intramembrane channel. It's a channel protein that connects the two different things. That connects two different spaces through membrane. There we go, brain. As this all pumps through, we're turning a chemical gradient energy into mechanical energy. And what do we do with that mechanical energy?
Use it to
Make more ATP.
make more ATP.
Yeah, we use it to make ATP. Sweet. So comparing, this right here, if you can draw it like this and this flow, you'll be chilling in a good chunk of the test when it comes to these types of questions. Because like the way that he is going to ask these questions is how things flow, what enters where, and then as we look at some of the other questions, we're going to start to look at kind of what sort of things block or stop at different points. And so we have this main drawing here, and then we can slowly add in those potentials like, okay, what blocks what here, and why does that happen? There's a lot of flies in Pennsylvania. We live next to a farm, and so we constantly have flies in the house now. Okay. Let's jump back into questions. ATP synthetase in the electron transport chain contains two functional units. One of these is an electron transporter, is a dehydrogenase component, or is a transmembrane proton channel.
Transmembrane proton channel.
Yeah, because what's it transporting?
Hydrogens.
Hydrogens, which are
That's transporting hydrogen. Does it have an electron transporter?
No.
No.
No, because the last complex to get an electron and pass it off to water is which complex?
4.
Perfect.
Four.
And complex 4 is made up of which two cytochromes?
A and
A and A3.
A3.
Perfect. Good job. Next question. Biosynthesis of ATP during oxidative phosphorylation can be blocked by which of the following? Increasing ADP concentration, FAD, blocking FOF1A TPase, or all of the above?
Logically, I want to say all of the above, but the FAD throws me off there.
Talk about it. Why does it throw you off? Or what does everybody else think?
I would say at least C for sure.
So C, we have a vote saying one person wants all the above, another person says blocking of the big long protein name. So we're talking about stopping ATP, right? So if we think about remember when we've talked about feedback and feed forward loops?
Mhm.
Mm-hmm.
When we talk, let's talk about a feedback loop, right? What does a feedback loop mean?
Basically, like you have enough concentration of X at the end, so you're gonna stop it from
So
creating
it's
more.
like A combines with B to produce X, and X comes back and say, hey, we have a bunch of this, let's stop. And it stops the production of more X once there's enough of it, right?
Yeah.
Yep.
So if we look at this question, let's say in the electron transport chain, our end goal is what? What are we trying to create?
ATP.
ATP.
ATP. So if we have, what do we put together? We have energy. Let's see, we have energy from the hydrogens plus the ADP plus the phosphorus to get ATP, right? So if we increase ADP concentration here, is that going to stop us producing ATP, or is that going to make us want to produce more ATP?
I guess that would make it want to be more.
Okay, so
More,
if we
yeah.
increase our ADP concentration, we want to increase our ATP concentration. When we talk about FAD, at any point, does, besides creating FAD using which complex takes FADH2 and turns it to FAD?
2.
Complex 2. At any other point, do we care about FAD, or does FAD interfere with anything else going on?
No.
Exactly.
Nope.
So FAD, so it's not going to go anything there. It won't be there. So our answer must be blocking this guy. And this is one of the ways that we learn. So this specific protein, this is what's present. This is part of complex 5. It's present in the inner membrane of the eukaryotic mitochondria, which acts as the power to the cell by synthesizing ATP. So this specific protein right here, or FOF1 ATP synthase, is just the longer name of ATP synthase, which is what helps make our ATP. So basically what this question is saying, which of the following making ATP is going to stop if what is blocked. If we block the ATPase, the ATP synthase, then we're no longer able to produce ATP. This is just more the chemically fun name that they use for it. Okay. This is another kind of in his book he has. Oh, did I close out his book? No, there it is. In his book, he goes through a lot of these where he just kind of goes through different things that can block. Here we go. Blockers of the electron transport chain. Retinone, antimyosin A, cyanide, carbon monoxide, these kind of stuff. All of these, I'm going to tell you right now, at least 1 or 2 of these are going to be test questions in some shape or form. This is a good chunk right here. Just these couple of things. Make a couple of flashcards and really memorize these ones. Know kind of where they like to block. Because he does like to at least throw 1 or 2 questions in of these guys. So biosynthesis of ATP during oxidative phosphorylation can be blocked by. Now we already talked about this is essentially the exact same question as this above, except a couple different answer choices. So we talked about ADP already. We talked about FAD. So our only thing left, and it can't be all of the above if those 2 are wrong. Last thing is our oligomycin. If I'm not mistaken, oligomycin is like a pesticide or something like that. Oligomycin. It's a naturally occurring one. It's a drug that people use. Strong antibacterial agents, often poisonous to other organisms, including humans. Yeah, so it's a drug that can be used to kill bacteria, but no good if we get it into our mitochondria. All right. Complex 1 electron transport chain transfers electrons from these. This should be bread and butter right here as far as questions.
C.
Yep.
C.
NADH to ubiquinone. Come on. From ubiquinone, where does it go to next?
Complex 3.
Complex 2.
Uh-oh.
Gonna go, remember, Elizabeth, what is complex 2? Where does complex 2 receive electrons from?
From the FADH2.
Yep, from FADH2.
Yeah,
Does
3.
Yeah. So we get the electrons here, transfer them to CoQ, FADH2, complex 2 transfers to CoQ, and then we skip to complex 3. So we don't go directly from complex 1 to complex 2. Both of these feed into our CoQ, or aka ubiquinone, and both transport to complex 3. Specifically in complex 3, where does it go to first? What's our cytochrome?
Cytochrome B.
Bam, cytochrome B, perfect. The more times we review that, the more times I make you think about it, the easier it's going to be to remember later on. Okay, here's another question about the stuff that stops stuff. Cyanide, oligomycin, and 2,4-dinitrophenol, or aka DNP, are inhibitors of mitochondrial aerobic phosphorylation.
Yep.
Which of the following statements correctly describes the mode of action of the 3 inhibitors? So the whole first chunk we can kind of skip reading. So these ones compete with O2 for cytochrome oxidase in complex 4. They inhibit the respiratory chain. Oligomycin inhibits the synthesis of ATP. Cyanide inhibits respiratory chain. Oligomycin and 2,4-dinitrophenol inhibit the synthesis of ATP. I'm not even really going to go, I don't want to go over each of these. Let's just go over the answer, because if going over the answer is going to be more beneficial in the long run. So cyanide is going to inhibit the respiratory chain. I'm going to pull up the book again, just like I said. So cyanide and carbon monoxide right here, it inhibits the respiratory chain. So it's that transporting that electron to that last step into the into the oxygen and to produce the H2O. And then oligomycin and the DNP, those are both going to inhibit the synthesis of ATP in general. I don't think that they give us. It might be in his PowerPoints that he shows those specific examples, because they should be in here somewhere. But yeah. You know, these guys, oligomycin and the 2,4 inhibit the synthesis. Cyanide inhibits the respiratory chain. It would not surprise me if a question like this, I could see him having this. I would star this one, because he'll have this exact same question, or one very similar, either like one of the parts of this question on the test. Okay. Jeez. Cytosolic NADH can be transported into the mitochondrial matrix by which of the following? So when we talk about cytosolic NADH, does anybody know kind of what that's in reference to? Why do we have NADH in the cytosol?
It's produced during glycolysis.
Yeah. So remember, we think about back to glycolysis. When we were creating, we used 2 ATP and then created 4 ATP for a net total of 2 ATP. As part of that process, we also made a handful of NADH. Now that NADH, we want to be able to use it, but it was created out in the cytosol. It needs to be transported into the mitochondria through these membranes. The way that we're going to do that is through the malate aspartate shuttle. Again, this is another one of those things you'll want to draw it out a couple of times. It looks way more confusing than it actually is. Basically, what it does is it uses malate and aspartate to shuttle or to transport, to carry the NADH and specifically the electrons across by doing some just kind of binding and converting different things to get the NADHs and the electrons that they were carrying across the membrane to get where it needs to go in order to be able to be used by the electron transport chain. So this whole, again, like I said, this is another one we want to draw out. Let me pull up Paint so we can make this look a little bit nicer, a little bit simpler. So we have our membrane. Here, actually, let me do this. I think there's a blank spot at the bottom here. You do this. Okay. We have our two carrier proteins, the malate alpha ketoglutarate carrier. If we had to guess, what two things does that carry or does that use to move across?
You gotta have malate and alpha ketoglutarate.
Yeah. And then we have the glutamate aspartate carrier. We need glutamate and we need aspartate. A lot of these different these chemicals, the malate, alpha ketoglutarate, glutamate, aspartate, they're very, very similar to each other. There's just one little detail that changes them and makes them a little bit different, which is why we're able to, using the NADHs, the NAD plus, those components, to kind of produce those. So we start off, we have here, this is, let's put this is our cytosol, and this is the mitochondria. So we have our NADH right here. And then we have malate. Malate is going to go across, trade spots with an alpha ketoglutarate. Now we have malate on this side. The malate will come full circle on this, and you'll kind of see how this connects. The malate is going to give the electron and the hydrogens to NADH. It's going to give it to an NAD plus. So then we have the formation of NADH. And by so doing, it turns into write it. It turns into oxaloacetate. Oxaloacetate is going to react with, remember we have in here glutamate coming across. Whoa. Okay. Aspartate, glutamate aspartate coming across. That's why it's called the malate aspartate, right, Shuttle? The glutamate interacts with the oxaloacetate. By reacting, it becomes the alpha ketoglutarate, which then turns the oxaloacetate into the aspartate. That aspartate goes across. Aspartate reacts with the alpha ketoglutarate. Alpha ketoglutarate turns back into glutamate. Aspartate turns into oxaloacetate. I got a little bit confusing because I'm trying to draw it out, but you can see everything is super, super close related, and it just flows. Malate turning into oxaloacetate. This step right here, this is the big step where we transport the electron across. Okay, so we're not actually taking the entire NADH and shifting it across. What we're doing is we're pulling off the hydrogen and the electrons associated with it, binding it onto the oxaloacetate to be able to transport it across nicely. And then NAD plus on the opposite side is then taking that hydrogen, the electron away again to convert malate back to oxaloacetate. And now you have the NADH across the membrane. So this is another one. Like I said, draw it out once or twice. This makes it look a lot more confusing because it has all the chemical structures drawn out. But it's basically like there's a big outside loop and then a smaller inside loop. If you understand the steps between these two loops, you'll be perfectly fine with all of the questions that you're going to get asked about this transport system. Where's my computer? Okay. Hopefully I didn't confuse anybody more with that. I wanted to draw it nicer, but wanted to show it's not as. I remember Dr. Sarkar, you can tell me if this is the same for you guys. He really never went super in depth about the shuttles and was just kind of like, hey, there are shuttles, and then moved on. So I don't know if you felt the same way or if there was any confusion there, but hopefully it's a little less confusing now, hopefully.
That's about right. From what I've seen so far.
Okay. Question 9. During a biochemical pathway, a shuttle system needs to transport electrons from cytosol NADH into the mitochondrial matrix, leading to oxidative phosphorylation, which of the following molecules carries the electrons in cytosol and itself is transported into the mitochondria. We have two choices, right? We have our malate alpha ketoglutarate carrier, and then our the aspartate glutamate, right? We're talking about going in. Malate goes in. Glutamate goes in, so it's one of those two. It's either the malate or the glutamate. Only choice that's here
It's malate.
is the malate. Does that make sense why it's malate?
Yes.
That's one yes. Any other yeses?
Mostly.
Yes.
Mostly? Erase all the stuff so it doesn't look as gross.
And it would be because the malate is the one that interacts with the NADH, right?
Exactly.
Okay.
So which of the following molecules carries the electrons from the NADH in the cytosol and itself is transported into the mitochondria, If we look at the full thing that's here, the only two things that are crossing the membrane are the malate and the glutamate. Those are the only two things crossing into the mitochondria. So that would have to be one of those two choices because those are the only two things going into the mitochondria.
So in the picture in his book, basically left side is the cytosol and right side is your mitochondria. And then yeah.
Correct, yeah. Yeah, this left side is the cytosol, where we have the NADH. This right side is the mitochondria, where we want the NADH to go. And it loops around, kind of converting, doing these quick conversions, passing different molecules, different atoms, different things around. And it changes the names of, like I said, like when we do the Krebs cycle, there's a whole bunch of little small changes that occur, and then they're called different things. So all of these, like the aspartate, oxaloacetate, malate, these are all involved within the Krebs cycle as well, which is why within the mitochondria, we have malate and oxaloacetate and aspartate and glutamate and alpha-ketoglutarate hanging around because the Krebs cycle is occurring within the mitochondria. So the body has the body was made or adapted or whatever you want to believe, so that it can use what is available to get the NAD plus, to get the high energy electrons that were made in glycolysis across. So it's using what's readily available to perform this action. Okay. Electron flow down the electron transport chain leads to a dangerous imbalance of potassium ions across the mitochondrial membrane. At any point did we talk about potassium ions?
No.
No, so immediately that's garbage. The transport of protons across the inner mitochondrial membrane from inside the matrix to the intermembrane space. That sounds plausible. The transport of protons across the inner mitochondrial membrane from the intermembrane space into the matrix. So that right there is from the intermembrane space into the matrix. Is that right? Do we go from the intermembrane space to the matrix, or the matrix to the intermembrane space?
Matrix to intermembrane space.
Yeah, matrix, we go from the most inside that matrix to the intermembrane space. So it's not there. The coupled synthesis of GTP. Do we use our GTP at all in our electron flow and the electron transport chain?
No.
No.
No. Process of elimination. Answer is B. I'm gonna keep kind of flying through these like this. If anybody has any questions or something is not clear or understood, please stop and let me know, and we'll come back and explain that. Give me 1 sec. Okay. Exergonic return of which of the following into the mitochondrial matrix results in ATP biosynthesis during oxidative phosphorylation? That's a lot of words to say what moves into the mitochondrial matrix to produce ATP.
Hydrogen.
A hydrogen. I know there was a question in one of the group me's about how complex 5 is both exergonic and endergonic. Do we want to talk about that for a sec, or do people feel okay with that?
I let Cheryl know that you brought this one up, so if you want to cover it, and then we can just go over it.
Yeah, put a description in group me, but super quickly. The hydrogen going through the channel releases energy, releasing heat. That's exergonic. And then the movement is then absorbed by the ATP synthetase, which ADP going to ATP is endergonic. So this is exer, and this is ender, and this whole thing is technically considered complex 5. So complex 5, that's why complex 5 is exergonic and endergonic, but it's mostly considered exergonic because more energy is going to be released than what is going to be absorbed and used. And that's the way all reactions are. If they're paired, if you have an endergonic and an exergonic reaction, and you pair them together to have reactions occur, there's always going to be more energy released than what is required. For example, ATP, you'll learn about this more in Phys 1. I think, Kristen, you're in Phys 1 right now, right?
No.
You're not? Okay. Within Phys 1, this is another little sneak peek. You'll talk about the myosin. You might have heard this from previous undergrad classes as well. You'll talk about myosin and actin binding chains within muscle fibers, within muscle cells. And so you have these actin and these myosin binding sites where everything will bind and then pull. And our ATP is broken and used to release this bond that actin and myosin have on each other. So then it releases, lets it kind of clock and reset, so then it can rebind and pull, and that was what makes our muscles move. When we exercise, what happens when we exercise? As we start to exercise, do our muscles heat up or do they cool down?
Heat up.
They heat up. Heating up or releasing heat is what kind of reaction?
Exergonic.
Exergonic reaction, right? So as ATP breaks, part of their energy is being used, energy is being absorbed and being used to rip off this portion and reset the myosin head. But because we were always going to have a greater exergonic energy release than an endergonic need, we get excess heat and our body produces more and more heat. So when we move our muscles, when we exercise, when we use ATP, we get the breaking of the bonds. Again, you don't need to know any of that, what I just talked about right there, about like the actin myosin binding, that kind of stuff. There's just a little sneak peek. You will see that very, very heavily in Phys 1. So just a little sneak peek, kind of what that is. That way, when you get there, you'll recognize it a little bit. These questions, Dr. Sarkar loves questions like this, except he'll have it like start at different points. I guarantee you that this or something very, very similar is a test question on your exam. Identify the correct sequence of electron flow in the electron transport chain.
It is an exam question. I will tell you that.
Guarantee it. So first things first that we look at, we always look at the beginning. A lot of times the beginning and the end, one of those two, sometimes those will be changed. And a lot of times we can tell correct answers right from there. In this one, everything's fine. So even NADH, CoQ, those are the same. The difference starts right here. So NADH right here to CoQ10. This right here, what's right here? How do we get from NADH to CoQ10? Through which complex?
1.
Through complex 1. That's the same. There, and there. So there's not the difference. From complex 1. There's 1, 2, 3. CoQ10. Our two little circles. From complex 1, we go to CoQ10. And then we go here. What's our first little circle in complex 3?
B.
Cytochrome B. Bam. B's our answer. We don't need to look at the rest of it. Unless we had another one that said cytochrome B. Then we'd want to look at the rest of it. But that's going to be your approach to answer these kind of questions. Check the beginning, check the end, and then go from the beginning and down and cross out and eliminate as you go. If electron transfer is tightly coupled mitochondria is blocked with anti-myosin A between cytochrome B and cytochrome C1. Where are cytochrome B and cytochrome C1 located?
Complex 3.
We have complex 3. Then which of the following will happen? Energy diverted from the cytochromes will be used to make ATP, and the pH ratio will rise. Electron transfer from NADH will cease, but O2 uptake will continue. All ATP synthesis will stop, or ATP synthesis will continue.
C.
C is the correct answer.
Yep, C.
Yep. And that's because if you think about it like a literal train, we have the tracks. The cars are going along the tracks. If one car stops, nothing else can continue going. We get a backup. The electrons get stuck. If the electrons are stuck, then they're not going to continue to go, to get dropped off, given to oxygen to be turned into H2O. And so that the whole process is not going to continue to happen. So the ATP synthesis is going to stop. In oxidative phosphorylation, aka electron transport chain, coupling of the electron flow through the respiratory chain and the ATP production are explained with which of the following? Ionic hypothesis, chemiosmotic hypothesis, conformational changes of the coenzyme Q, or chemical hypothesis. This is just kind of more of like a memorize-y question.
Would it be chemiosmotic because we're talking about the concentration gradient of hydrogen, maybe? Oh,
That's correct, yeah. So this is more just kind of definition-wise, and honestly, we don't really need to know the other definitions. We just want to know this one. So in the chemiosmotic hypothesis, a transmembrane ATP synthesis. This should be TACE. Whatever, synthesase, whatever. It's chemical, it's spelled differently. Is central to convert energy of spontaneous flow of protons through them into chemical energy of ATP bonds. So just know that electron transport chain is the chemiosmotic hypothesis. In the glycerophosphate shuttle, glycerophosphate receives a pair of electrons from NADH and donates this pair of electrons to blank. So we had the malate and aspartate shuttle, and now we have the glycerophosphate shuttle. The way that this one works is we take the NADH, We have glycerol 3-phosphate, dihydroxyacetone phosphate, which these are part of our glycolysis. And these are used to transport electrons across the membrane, and it gives them to FADH. The question is, receives a pair of electrons from FADH and donates the pair of electrons to, kind of a gross question, the way that it's worded. The answer is FAD, because it gives them to FAD, and then FAD becomes FADH2. So I don't love this question. I think this question is kind of, I don't like it the way that it's, when you include both of those like that, it's kind of gross. Because I think FADH2 is potentially a fair answer as well, but it's fine. So the glycerol phosphate shuttle, it's NADH, it gives it to our FAD into the electron transport chain, and then it goes directly into that electron transport chain. This is just another way that NADH, those electrons, can pass and go into the electron transport chain, besides the malate aspartate shuttle. Okay, just remember, if any questions, stop and let me know. In the malate aspartate shuttle, glutamate is converted to blank in the mitochondria, while aspartate is converted to blank in the cytosol. So remember, we have our two carriers, and we have them coming in. And there's the two different circles, right? So glutamate gets converted. So glutamate, I'll just pull up the thing. Glutamate's coming in, right? Glutamate gets converted to alpha ketoglutarate. Here in the inner circle, it gets across, converted back to glutamate, all the way around. So this first part, glutamate, is converted to alpha ketoglutarate. So right away, we know that that's the case. B is our answer because no other choice has alpha ketoglutarate. I do feel like most of his tests, he'll typically have two answers that are at least similar, like two answer choices will have alpha ketoglutarate as the first one. But yeah, again, this is just being able to draw this out. Yeah, just like I said before, if you can draw this out, especially drawing it quickly, you don't have to write everything out. We could even draw it more broken down, kind of like this. Malate goes across, malate, oxaloacetate, aspartate, aspartate goes across, aspartate, oxaloacetate, malate, alpha ketoglutarate, glutamate across, glutamate, alpha ketoglutarate. If you can draw it like this a couple of times and as long as you understand what it is saying, this takes 10-15 seconds to draw super quickly on a piece of scratch paper, and then you can answer any questions about the malate aspartate shuttle that the test will throw at you. Okay, one of the electron acceptors in the electron transport chain is blank.
NAD plus.
Yep, NAD plus because it's the only one that is not fully saturated and it can accept electrons because it already has a positive. All of these other ones already have their hydrogens and everything attached. There's no reason for an electron to bind to them. One of the protein components of the electron transport chain is blank. One of the protein components of the electron transport chain is blank. Remember ubiquinon? I said that ubiquinon was very, very closely related to a vitamin. Anybody remember which vitamin?
Thiamine?
No.
Oh, vitamin K.
Yeah, vitamin
Okay.
K. Good job. So ubiquinon is very, very closely related to vitamin K. Do you remember when we talk about vitamins and minerals, right? What do we call those?
Cofactors.
There's cofactors. And what else?
Enzymes.
Coenzymes. Vitamins specifically, are vitamins cofactors or are vitamins coenzymes?
Cofactors.
Factors.
Cofactors.
You watched the videos before. Remember the stupid thing I said that? Remember? Did anybody ever watch Fear Factor?
Yes.
Okay,
You
so
said
to
it's
go on
very
Fear
metal.
Factor,
Cofactors are metal.
you have to be pretty metal. Our metals are our cofactors. Our vitamins are our coenzymes. So vitamin K is a coenzyme. Metals like iron, magnesium, copper, potassium, sodium, calcium, all of those are cofactors. Any of our vitamins, vitamin A, B, all B vitamins, C vitamins, vitamin C, D, all of those are going to be coenzymes. Okay, NAD plus. Is NAD plus a proton or not a proton, a protein?
No.
Not
It's
a
also
protein.
derived from a vitamin, right?
Yep. Do we remember what vitamin? It's niacin, which is a B vitamin. Does anybody remember what B vitamin it's derived from?
B3? 1 or 3, I can't remember which one.
Niacin.
All rich nudists played pickle ball for centuries. Vitamin 1, B1, thiamine, B2, riboflavin, B3, niacin. Vitamin B3. Just a little review. These will come back a lot in. You want to know these pretty well for Biochem 2, as well as try 5? Try 4 or try 5. You have a clinical nutrition class that hits vitamins really hard, and then boards as well. Loves vitamins. And then flavin nucleotide, flavin, riboflavin, B2, that's our FAD. So complex 4 is the only one that's actually a protein. Oxidation of one molecule of which of the following compounds will produce the maximum numbers of ATP. This one's kind of a gross question. I don't feel like you ever really talked about this. The answer is sucrose. Think
Because it's a
that
disaccharide or?
yeah,
Okay.
because sucrose is glucose and fructose. I don't see him really asking that specific question. It's kind of a gross question. I don't like that one. Okay, oxidative phosphorylation requires the transfer of electrons donated by which of the following? Again, these are the kind of questions that I feel like can be a little bit tricky. So think, which of the following, which of these four currently has electrons that it will in the future donate?
NADH.
NADH.
The NADH. Oxidative phosphorylation takes place where?
Mitochondria.
Mitochondria.
Mm-hmm.
Just to make sure that we're clear. So oxidative phosphorylation equals the electron transport chain. It's saying the same thing. So whenever you see oxidative phosphorylation, think the electron transport chain. It's saying the exact same thing. What happens in the cytosol?
Glycolysis.
Where does the Krebs cycle take place?
Mitochondria.
Nice. The electron transport chain is localized in
Mitochondria.
mitochondria, specifically where within the mitochondria? Where are the proteins of the electron transport chain located?
The intramembranous space.
Between that and the matrix, right?
Yeah, so the mitochondria
Yeah.
has two membranes, right? It has the outer membrane, it has the inner membrane. So we have the outer mitochondrial membrane and the inner mitochondrial membrane. So the inner mitochondrial membrane, which separates the intermembrane space from the matrix. So the matrix is at the very, very middle. Then you have the inner mitochondrial membrane, and then you have the intermembrane space between the outer and the inner membranes. He likes, especially on the, do you guys have one more exam before the final, or is your next exam the final?
I'm pretty sure we have one more before the
One
final.
more and then the final. Yeah.
Okay. I don't remember if it was on the third exam or on the final exam. He likes to ask questions about like the inner membrane. A lot of times he'll use, instead of like the inner membrane space, he uses intermembrane space. He'll have on there intermembrane space and intramembranous space. Like he'll do stuff like that. That's kind of tricky. So just make sure that you know kind of what's what, that it's the intermembranous space between the outer and the inner membranes of the mitochondria and that kind of stuff. You heading to bed? Yep. Yeah, give me 1 sec.
So cute.
He's the best. Okay. The energy released by electron transport chain system produces ATP by which of the following?
Oxidative phosphorylation.
Oxidative phosphorylation. The 4 protein complexes that are involved in the transfer of electrons from the reduced content to the oxygen are localized in. This is what I just said. Did anyone want to answer?
It's
Inner
the inner mitochondrial membrane.
Yeah, the inner mitochondrial membrane.
Yeah.
I was like, we just talked about this. Okay. Uncoupling of mitochondrial oxidative phosphorylation. What does uncoupling mean?
Bond breaking.
Yeah, uncoupling, or can like breaking parts of it, undoing, that sort of stuff. So breaking down of mitochondrial oxidative phosphorylation halts mitochondrial ATP formation but allows continued O2 consumption. Which of the following? Halts all mitochondrial metabolism, slows the conversion of glucose to pyruvate by glycolysis, allows continued mitochondrial ATP formation, but halts O2 consumption.
Halts all mitochondrial metabolism.
So let's stop and
Question.
let's think Krebs cycle, right?
Mm-hmm.
Where does the Krebs cycle take place?
Your mitochondria.
Inside of the mitochondria still. So that's still part of mitochondrial metabolism, right?
Mm-hmm.
So even if, and as well within the Krebs cycle, right, we're bringing oxygen in and we're pushing out and producing CO2. So the answer for this question is going to be that it halts mitochondrial ATP formation, but allows continued O2 consumption. So if just the electron transport chain breaks down, we're going to stop that production of ATP, but things before it are still going to be occurring. Again, this is one of the questions that the exact reasoning why I could see him putting a question like this on the test. Like the exact reasoning as to why I personally feel goes a little bit above our pay grade. Just like they're getting deeper into like the nuances of why and stuff, I don't think would be the best use of our time. But this is a question I would put a little star there and come back to it at some point, like when you're going through prep and right before the test. Just to kind of, with these kind of questions, he'll throw a couple of these on there that are just like kind of like the difference makers between, you know, that low 80s grade up to getting up to a 90 and pushing up above that. So these kind of questions are the ones that typically do that.
Yeah, I guess I still don't even understand what's uncoupled there, but I guess it's you're inferring that maybe it has more to do with like the separation of the Krebs cycle from the electron transport chain.
So uncoupling, so the uncoupling proteins are transporters present in the mitochondrial inner membrane. They mitigate a regulation discharge of the proton gradient that is generated by the respiratory chain to serve functions such as thermogenesis, maintenance of the redox balance, or reduction in the production of reactive oxygen species. So the uncoupling proteins are the transporters within the mitochondrial inner membrane. Look back at the question.
That helps.
Uncoupling, so the uncoupling proteins are those transporters. But also at the same time, if you say uncoupling, it literally does mean like breaking down or halting, kind of stopping a process. So that's why I say this is one of those questions where I don't love the question itself, and I don't feel like it's a super. If he went any deeper than this, it'd be an unfair question, because like I said, I think it's a little bit above our pay grade. So when we talk about uncoupling of the mitochondrial electron transport chain, it's literally kind of like the breaking down of it. Like the individual components get, if you uncouple them, you kind of separate them so that it's not functioning as it should. So if we separate the proteins within the chain, they're not going to continue to pass the electrons like they're supposed to. And then we don't get ATP production at the end because we're not allowing the process to actually occur. So
on the most basic level to like remember this question, it stops the electron transport chain, but allows Krebs cycle to continue.
Yeah, and we'll still be using oxygen within part of the Krebs cycle. So yeah, again, one of those kind of recognize the question. So if he does ask this question, it'll be this exact same question. So okay, back to this kind of stuff. What is the effect of antimycin A on the electron transport chain? And so it's going to block ATP synthesis. Specifically, we'll just look again because the more times that we see something, the more likely we are to remember it. Specifically, it stops the transport between cytochrome B and cytochrome C1, which is located in which protein complex?
3.
Complex 3.
In complex 3. So another way he could word this question is, which of the following does antimyosin act on? And the answer would be complex 3, because it blocks between cytochrome B and cytochrome C1. What is the metabolic fate of oxygen in the electron transport chain? It's first converted to carbon monoxide and then to carbon dioxide. Oxygen is reduced to form water. Oxygen is converted to carbon dioxide, or it produces acetyl-CoA.
Water.
Yeah, oxygen is reduced to form water. Of all of this, glycolysis, Krebs cycle, and electron transport chain, where is a carbon dioxide produced?
Krebs cycle.
Within the Krebs cycle. Specific, what's the name of the type of enzyme that produces carbon dioxide?
It is. It's either, I think it's the alpha ketoglutarate dehydrogenase, or it's isocitrate dehydrogenase. One of those two. I'm looking at my drawing.
I keep thinking of, are there like
you remember the-
decarboxylases?
Yep, the decarboxylase. These guys, the decarboxylation enzymes, are the ones that remove a carboxyl group and releases carbon dioxide. So within the Krebs cycle, we have our decarboxylase enzymes, which are the ones that are going to be releasing the carbon dioxide. Again, that's more like thinking future, the final, when you have a little bit of everything on that test. So the decarboxylase enzymes are where the CO2 gets produced.
It's like I need to add some stuff to my drawing.
Yeah. What is thermogenin? So I'm just going to go ahead and give you guys this one. We'll talk a little bit about it. So this is an uncoupling protein found in brown adipose tissue. Has he talked to you guys? Do you guys recognize at all when you mentioned brown adipose tissue?
Dr. S has talked about it in BCT.
Sarkar went over a little bit, I think, on Friday with our one-on-one, but I don't know if he went over it in the group lecture.
Yeah, so brown adipose tissue, it's more common. We mostly talk about it when we talk about infants. Infants have a lot more brown adipose tissue. But so the brown adipose tissue, so it's a type of body fat that regulates your body temperature, specifically in cold conditions. So it activates right before you start to shiver, and brown fat also burns calories and stores energy. So you have a small percentage of brown fat in your body compared to white fat. Within an infant, for example, infants tend to have a lot more brown fat than they do white fat. So that's why infants are able to better regulate, they're very quickly regulate the temperature control, because if an infant gets cold, it's much more detrimental to their health than if like a full-grown person gets cold. So thermogenin is a specific protein it's an uncoupling protein found in the brown adipose tissue. As far as its function goes, don't remember its exact function. Thermogenin function. So it functions as a proton transporter, allowing the dissipation as heat of the proton gradient generated by the respiratory chain, and thereby uncoupling oxidative phosphorylation. So okay. This is actually going to be a better explanation. It clicked again on my brain. Better explanation of the uncoupling. Think of it like in our whole process, right, of one, two, three, four, and then here. The uncoupling process, essentially what that's referring to is it separates the wind turbine like this. When we're talking about the uncoupling, in this case, especially for the brown fat, it takes off and it blocks and stops the ATP synthase portion. So then what it does is all of the energy from the hydrogens flowing through here, instead of their, because remember the hydrogens flowing through is exergonic, and then the ATP formation is endergonic. So instead of there being ATP formation within the brown fat, it just leaves the exergonic release of the hydrogens moving across in order to heat up the body. And that's also why when we look up here, uncoupling of it, this is uncoupling, so it's going to stop the ATP synthesis, but the rest of the reaction occurs.
Awesome. Thank you.
That's why we keep flowing through it, because eventually we figure things out. So thermogenin, if you ask about thermogenin, that's its job. So you could even ask it in the context of this. So you could even say the function of thermogenin within brown adipose tissue is its colon. And you could, this exact answer choice where it heights the mitochondrial ATP formation but allows continued O2 consumption, this answer choice would fit perfectly as well with any question asking about thermogenin. Any questions about that? I know we kind of jumped around a little bit there.
All good here.
Okie dokie. Which form of coenzyme Q is used to accept electrons from complex 1 in the respiratory chain?
Oxidized?
It is oxidized. I feel like most of the time, either oxidized or methylated. Those are going to be your two go-tos. I don't remember this being hit on super big in the book either. I'm sure it's probably like a one-liner somewhere. But yeah. Another question, just to look at where you can't really work your way through it. It's just a memorization game on that question. Which is one of the protein components of the electron transport chain? We already had a question just like this. But that complex 2, because it's the only one that's actually a protein. The other stuff are vitamins and such. Coenzyme Q, most likely a vitamin. Vitamins, are they coenzymes or cofactors?
Enzymes.
Yeah, coenzymes. Good. Which of the following can be an uncoupler of the electron transport chain?
Diphenyl.
Yep. Which of the following components of the electron transport chain contains flavin nucleotides? I think when we talk about flavin, right? Riboflavin, the flavin, does that correlate more to FADH or NADH?
FADH.
Or FADH2, right? So which of these
Mhm.
complexes has to do with
Complex
FADH2?
2.
Complex 2.
Nice.
Which of the following compounds receives electrons from both complex 1 and complex 2 in the electron transport chain?
Complex 3.
Before they go to complex 3, where do they go first? This is because that
Oh,
complex 3
coenzyme
is
Q.
technically correct, but coenzyme Q is more correct.
Gotcha.
I hate
I
when
thought the
teachers
same thing.
I hate when teachers say that, like, but what's the best answer choice? It's stupid. I hate it. But here we are.
But
So
technically, they are going to complex 3 eventually.
yeah, technically they're also going to complex 4 and to the well, not the ATP
That's
synthase,
also
but
true.
technically this is probably a really good lesson of demonstrating thought after the fact. If you had
Mm-hmm.
clicking through the test, you're going really quick, and you clicked complex 3 and moved on. When you go and you meet with Dr. Sarkar, posted it, or another teacher in a similar situation like this, that is where you need to look at it and then demonstrate your knowledge before the teacher says anything. Read the question. I would read the question, and I would immediately say to the teacher, "Oh, I see what I did. I misunderstood." Because technically, yes, technically they go 1 and 2, and they both go to complex 3. I didn't realize that coenzyme Q, because coenzyme Q was before that, and kind of like quickly get in a word of explaining why you chose your answer before the teacher goes on and tells you why the answer is the correct answer that it is. If the teacher allows you to do that, there's a good chance that you can get some pointage, even like half points back on stuff. Just a little tip, especially teachers.
Sarkar's ever given half points on anything.
Yeah. Dr. Sarkar is a little bit of a tough nut to crack, but a lot of other professors, especially in like physiology, systems pathology, those kind of courses, you can get points back if you can demonstrate your critical thinking of like why you thought the answer that you picked was correct. Yeah. Which of the following does not participate nor is a component of the electron transport chain?
Coenzyme A.
Coenzyme A.
Yeah.
Where's coenzyme A? Where does the coenzyme A come from?
Krebs cycle. Yeah.
Yeah, from the Krebs cycle. Which of the following enzyme catalyzed reactions of the citric acid cycle donates electrons to the electron transport chain? Oh, sorry. Give me 1 sec. My cat is stuck on the ceiling. Come here, you. Come here. I got you. Oh. Okay.
Himalayan.
We adopted her. She looks very Siamese, but she's a mutt.
Definitely got some Himalayan in there with that fur.
Go. I don't know how she got up there. Okay. The answer is the succinate dehydrogenase catalyzed reactions. This is kind of combining our knowledge of the Krebs cycle to the electron transport chain based on which steps of the citric acid cycle are producing our NADH or FADH2. So that's why make sure you review your Krebs cycle as well and have that in your back pocket. Does that make what I said? Does that make sense? Kind of what we're doing there?
Yeah.
Mm-hmm.
Okay.
That's where it produces NADH, right?
Yeah.
Okay.
There is a little bit of a mnemonic. If you didn't watch my other videos on it, let me re-emphasize it here. pull it up. Here we go. Within the Krebs cycle, if you can remember the steps of the Krebs cycle, I am the most important, meaning that under the isocitrate, the alpha ketoglutarate, and the malate stoxylacetate, that's where the NADHs are produced. And then actually the answer to this question is the succinate to the fumarate is where FADH2 is being produced for this question. So there's a couple of different helpful mnemonics, like for Krebs cycle, like couples cuddle insanely often after special sex for marriage obligations. Just kind of silly things like this that help remember. So I mentioned these in like the other videos and stuff that I've recorded, but in case you hadn't seen that before. Okay. So the succinate dehydrogenase should be, yeah, on our Krebs cycle. Where'd they go? Oh, come on. Our succinate dehydrogenase produces not NADH, but it produces FADH2, which gives us electrons. Okay. Which of the following is not a respiratory chain protein?
Coenzyme Q because it's not a protein.
That is correct. Which of the following are false about oxidative phosphorylation?
It involves oxidation of coenzyme Q.
That is correct. Because we look at this, so our previous question talked about, where is it? The form of coenzyme Q that is used to accept electrons from complex 1. It is already oxidized, so we don't need to oxidize it in order for it to work. It is already oxidized. Which of the following are true about oxidative phosphorylation? He loves these questions.
Yes, he does.
Free energy available from electron flow through the electron transport chain couples to ATP biosynthesis. That's true. It involves flow of electrons through a series of membrane-bound components. That is also true. Immediately, if two of them, I'm like, yeah, those are both true. I'm clicking all of the above. Then double check the last one. Transmembrane protein gradient is generated during electron flow through electron transport chain. That is also correct because we're making the gradient with the hydrogens. Which of the following molecules serves as the electron acceptor in the electron transport chain? I think we already had this exact same question.
Yeah, I think we did. NAD plus.
NAD plus.
Because it's the only one that can accept electrons. Which of the following statements about the chemiosmotic theory is correct? Just remembering that definition. Coupling of electron transport chain and oxidative phosphorylation will halt ATP biosynthesis. That doesn't tell us anything about the chemiosmotic theory. The membrane ATP synthesis has no significant role in the chemiosmotic theory. That's the whole point of it. Electron transfer in mitochondria is accompanied by an asymmetric release of protons on one side of the inner mitochondrial membrane. That talks about our release of the protons going across the membrane, all that sort of good stuff. And that is our chemiosmotic theory. Does anybody need to review what the chemiosmotic theory is? So if you see it, make sure you look for the question that's talking about, look for the answer choice that's talking about something to do with that proton gradient and stuff moving across. This is almost the exact same question. The answer choice is the same. Just a couple other incorrect answer choices, I guess. Which of the following statements is true about using an uncoupler in the electron transport chain?
ATP synthesis is blocked.
Yeah. What about oxygen?
It will continue.
What is the name of the protein? The name of the enzyme that is found in brown adipose tissue that uncouples?
Thermogen.
Thermogenin. Yeah, thermogenin. Good.
Thermogenin.
Brown adipose tissue, is it more commonly found in adults or infants?
Infant.
Good. Which statement is not true of electron transport? The electron transport chain oxidizes reduced cofactors. Cytochrome C has a higher reduction potential than FADH2. Electrons flow from redox centers with a lower reduction potential to redox centers with a higher reduction potential. The mobile electron carrier coenzyme A is located in the inner mitochondrial membrane. Remember, the question is asking not true.
Well, the electron transport chain doesn't have anything to do with coenzyme A.
Exactly. A lot of times questions like this, people get really freaked out because there's a lot of words. And I'm gonna be honest, reading these for the first time straight through, I was like, I would have to read that another time or two to figure out what the heck it's talking about. Like to actually like understand what the answer choice is saying. But look for those keywords where it's like, oh, this thing has no idea, or like this right here, I've never heard of this before, or like this doesn't, has to do with something else. And if it's not true, this question, that helps you answer these questions a little bit easier. Okay, almost there. Within the electron transport chain complex, blank represents the entry point for NADH, while complex blank represents the entry point for electrons of NFADH2. Which complex allows for NADH?
1 and 2.
1 and 2. Both complex 1 and 2 gives their electrons to what?
CoQ.
CoQ.
What is another name for CoQ?
Ubiquinol.
It is most closely related to what vitamin?
K.
Okay.
In what form is CoQ active?
Oxidized.
It's oxidized form. Good. CoQ transports the electrons to what cytochrome?
3.
B.
B.
Cytochrome B. What is the name of the chemical that blocks electron transport between cytochrome B and cytochrome C1?
Endomycin A.
Yeah. That made my brain hurt. Okay. What carries electrons from complex 3 to complex 4?
B.
Enzyme Q.
Answer
C,
A.
right?
Yeah, cytochrome,
cytochrome
C. Remember, because coenzyme Q is from 1 and 2 to 3. Cytochrome B is within complex 3, so that's complex 3. Cytochrome B transfers it to cytochrome C1. Okay. And then C1 to cytochrome C, and then it goes to cytochrome A in complex 4, and from cytochrome A it goes to what next?
A3.
Cytochrome A3, and then from A3 it produces what?
Water.
Oxygen, water.
It binds to oxygen and produces H2O. Good job, everybody. Great job.
Awesome.
We did it.
I think what's really, really cool about this is like when answering these kind of questions, remember at the beginning, I felt like we kind of struggled along through it a little bit. And then as we get further and further along, it's like, oh, okay, I know what's going on. Does anybody have any residual questions, concerns, anything else that you want to talk about before we log off for tonight?
Can you go back up to question 14? I just didn't finish typing out the definition of the chemismodic hypothesis.
There you go.
Yeah.
Okay, so this week, here's the plan. Tuesday, we'll plan on doing our normal time. I will log in. I'm gonna check and see if I can do. Are you guys okay if we do a little bit earlier still?
That's fine.
Does that work a little bit better for people?
Yeah.
Because I'm thinking, so
Yeah.
I would love on Tuesday to log in and do like 6 o'clock Central Time. Would that work for you guys?
Yeah.
For me.
Okay.
That's fine
And
with
then
me.
Thursday, I'm opening up some time. I already opened up some time on Penji. So I think we're going to do a group session starting at 2 p.m. Central Time. So I'll plan on logging in around 2 p.m. Do you guys have anything supposedly going on at that time?
We've got FDI front like
potentially.
until 2.
Until 2 o'clock?
Yeah. Well, 1:50 technically.
Okay. Do you want me to are you guys comfortable with jumping into biochem right after that? Or do you want me to push it back an hour or half an hour?
I mean, I'm good with jumping in. I may be like jump from east to a different place on campus to do it, but
Yeah. I'll plan on we'll plan on 2 o'clock for right now. If anything changes with that, or if we need to push it back, just send me a message and communicate with me. I think with this, we should be able to get fairly caught up with the questions and stuff, you guys, just because you guys have, looks like, were all of these quiz questions? Did you guys just have a lot of material this week?
We had TCA and activated phosphorylation. And then this upcoming week we have, I don't remember what it is because he hasn't launched it yet, but I know what it is based
The
on.
Glycogen metabolism, gluconeogenesis, and the pentose phosphate pathway. Have you guys already had those?
No, those are coming up. I just sent them to you.
say I think at least glucogen metabolism, that one, and the gluconeogenesis, I think both of those are this week.
Okay, sweet. So I think we can go ahead and hit those. We should be able to get both of those done for sure this week, and maybe even potentially get ahead with the pentose phosphate pathway. And then if we have a little bit of extra time, we can maybe jump back to the Krebs cycle questions, or to maybe another unit that, if people feel like there was a unit that they didn't do super hot in, we can go back to that and review that. So that way, when it comes up to the final, we can be a little bit more prepped for the final exam.
Sounds great.
We do have a BCT exam Thursday as well.
Oh, you do?
Oh yeah, we do, don't we?
Yeah.
Is that scheduled or is it open all day?
It's
I
open
think
all
it's
day.
open all day. Asynchronous.
Okay. I'll probably use on Thursday. We can probably just go ahead and use that time. I'll see. I will do questions if there is some conflicts or people are freaking out about the BCT exam. Just let me know, and we can either reschedule, or I can do the session for 2-3 people if they show up, and make sure it gets recorded. So I'll we can decide that as we get a little bit closer to there to Thursday. Sound good?
Yep.
Great.
Sounds good.
I appreciate it,
Thank
Drake.
you.
Okay, everybody. Have a great
Thank you.
night. Thank you for stopping in. I'll see you
Thanks,
later this week.
sir.
See you later.
Thank you.
Thank
See
you.
you next week.
Thank you.